Hi everyone. Today I'm going to talk about Alpha-1 Antitrypsin Deficiency, which is a genetic form of COPD. So COPD, or chronic obstructive pulmonary disease, is a very common pulmonary disease, usually attributed to smoking or environmental pollution. However, Alpha-1 Antitrypsin Deficiency is a hereditary cause of this disease. It's widely underdiagnosed because usually COPD is just chalked up to smoking, and so only about 10% of cases are diagnosed, and that's usually only if you're not a smoker at all, but you exhibit symptoms of COPD. Let's talk about the genetic mechanism of exactly how this disease works. So, Alpha-1 antitrypsin is a protein that circulates in our blood and sort of protects our lungs from a natural process that would otherwise degrade them. The gene which encodes it is called SERPINA1, which is found on chromosome 14, and this stands for serine proteinase inhibitor A1. It's called this because it was one of the first serine proteinase inhibitors, which is a large superfamily of proteins. So it was given alpha-1 as its sort of the archetypal form. You can see on the left here, this is a diagram of the protein structure, giving an example of a normal M allele, which is how we denote wild type. There are many mutant alleles which can cause alpha-1 antitrypsin deficiency, but one of the most common, well-documented, and severe is considered the Z allele, which is characterized by a point mutation at the 342nd residue of this protein, where glutamate is substituted for a lysine residue. So how does this work? When the Z allele is found and this gene is transcribed, hepatocytes in the liver secrete this malformed version of alpha-1 antitrypsin, which sort of agglutinate in the hepatocytes and never leave and circulate in the bloodstream at a sufficient level to do what they're supposed to. In a normal MM individual or a homozygous wild type, alpha-1 would be secreted by hepatocytes into the bloodstream, reach a serum level of between 1 and 2 grams per liter, and then eventually make their way to the lung, where they bind to neutrophil elastase, which is really the culprit of the disease here. Neutrophil elastase is a serine proteinase whose function is normally to help fight off infections, as it's secreted by neutrophils and will cleave membrane proteins in invading bacteria. However, neutrophil elastase can also cleave bonds and important proteins in the lung tissue, which I'll talk more about later. So if you are homozygous for the ZZ allele, or you're a ZZ alpha-1 antitrypsin deficient, then you will not be able to produce enough of this protein to actually inhibit neutrophil elastase, and your plasma or your serum levels will be too low to detect the protein. So what exactly does this cause when neutrophil elastase is damaging the lungs? Well, this is kind of how we diagnose and characterize COPD in one respect. You can see on the left here we have chest x-rays. Here's a normal one with a nice shapely diaphragm, as opposed to this COPD with emphysema, where you have elongated lungs. It's much, much darker air spaces, showing sort of overdistension and increased air here, and blunted costophrenic angles. You can also see a transverse CT over here, a normal one versus an emphysematous one. Another way we can look at the consequence anatomically of neutrophil elastase kind of going and being destructive to lung tissue indiscriminately is histology here, where we are looking at sort of a cross-section of alveoli. Here is alveolar epithelial tissue over here. This large white space is actually the lumen of an alveoli or other bronchi over here. And so we see this is over-enlarged, and the alveolar walls are kind of malformed and thinner than they otherwise would be. So what exactly are we talking about here when we're talking about anatomical destruction of lung tissue? Well, specifically neutrophil elastase will break down elastin and collagen, which are proteins found in the extracellular matrix of lung tissue. These extracellular matrix proteins give the lung elasticity and recoil, also called lung compliance. The greater the lung compliance, then the more the lung will expand given a breath, which sounds like a good thing. But if a lung is over-compliant, you will actually have airway obstruction, which is a big characteristic of COPD. You can see over here there's an example of some bovine collagen being pulled between two forceps, and you can kind of imagine how that would be pulled and have like an elastic characteristic to it. So in alpha-1 antitrypsin deficient individuals, the elastin and collagen of the extracellular matrix of their alveoli, and therefore the parenchyma of their lungs, become broken down, and they lack elasticity and they become overcompliant, which is what we're seeing in this example here. there's too much air, it's too floppy, and these epithelial cells are too thin. And over here, again, we're seeing the same thing, sort of overcompliance and emphysema. COPD is characterized by a wide variety of additional factors. However, the emphysema seen in alpha-1 can be distinguished as pan-ACNR as opposed to centra-ACNR, which is an important distinguishing factor. Testing usually, however, is done in a combination of pulmonary function tests and serum level tests, eventually culminating in a genetic test to finally discover exactly which alleles you have that are making you deficient in alpha-1. So treating alpha-1 antitrypsin deficiency, there's not a lot we can do for COPD in general. It's mostly supportive, as it's a degenerative, progressive disease that cannot be reversed. Alpha-1 is the same deal. There's a lot you can do to support individuals, but you can never reverse the damage done to the lung. One thing specifically for alpha-1 as opposed to the rest of COPD is something called augmentation therapy, which was first implemented in 1987. Essentially what this is you are given a weekly injection of exogenous human alpha-1, which is derived from plasma donation. And you need to get this injection weekly for the rest of your life to essentially stave off the destruction caused by neutrophil elastase. It won't reverse any of the disease progress that has already happened, but it will prevent it from progressing. However, it's not clinically proven to have consequential clinical outcomes in many respects and in many of the ways that we use to characterize COPD. So that's an overview of alpha-1, a hereditary form of COPD, and the genetics and mechanism behind it. Thank you.